Hi, everyone. We're going to start session 3. So my name is Yuqing. It's my honor to be here to introduce you the session 3 MMF applications for long-acting injectable drug products. So I'm a staff fellow at the Division of Quantitative Method and Modeling, DQMM, Office of Research and Standard ORS, and Office of Generic Drug CDER FDA. So in order of presentation, our first speaker is Dr. Andrew Babiskin, who currently holds the position of team leader for the quantitative clinical pharmacology team in the DQMM ORS OGD CDER FDA. And you will be hearing from his presentation on regulatory utility of MMF for development of LAI drug products. And the second talk will be delivered by Dr. Tonglei Li. Dr. Li is a professor and Ellen Chow Endor Chair in the Department of Industrial and Molecular Pharmaceutics at Purdue University. And you'll be hearing from his presentation on bridging critical quality attributes and system exposure of LAIs by multiphysics simulation. And the third speaker is Dr. Andy Hooker. Dr. Hooker is a professor of pharmacometrics at Uppsala University, Sweden. And he is also a co-developer of a number of software programs, including Expose PSN and the Optimal Design Program, POP-ED. And he will be giving the talk on model-based bioequivalence methods serving as MMF for LAI drug products. The fourth speaker, Dr. Daniela Silva. Dr. Silva is a scientist too at Simulation Plus, and she will be giving the talk on model master file in the context of LAI. And we also have a 30 minutes panel discussion, and we will have Dr. Partik Saha and myself to moderate the session. Dr. Partik Saha currently heads the Global Pharmaceutics Groups in drug product development that is responsible for integrated biopharmaceutics support across dosage form at GSK. And we also have additional panelists to join the panel. They are Dr. Khondoker Alan, who is currently working as a senior staff fellow in the DQMM ORS OGD in FDA. Dr. Murray Ducharme. Dr. Ducharme, he is a president and CEO at Learn Confirm in Canada and a professor at University of Montreal, Canada. And Dr. Rebecca Moody, who works in the immediate office of the Office of Drug Product Quality 2 at the FDA. So I'd like to welcome everybody to session 3, and I'd like to welcome you to join Andrew's presentation on regulatory utility of MMF for development of LAI drug products. So Andrew will be presenting virtually, so please play his video. Thank you.
Good afternoon, everyone. My name is Andrew Babiskin, a team lead within the Division of Quantitative Methods and Modeling. I apologize that circumstances did not allow me to present live today, but I'm still glad to open this session on the applications of the model master file for long-acting injectable or LAI drug products by providing a regulatory perspective. Through the 2 days of this workshop, you're going to hear a lot of cases of model master file for PBK modeling for both locally acting and oral drug products. But as you'll see in my talk in this session, the complex circumstances around LAI bioequivalence also lends itself to cases of population PK modeling to augment and support the in vivo PK bioequivalence studies. Before we get into the rest of the presentation, this slide is to remind you that this presentation reflects the views of the author and should not be construed to represent FDA's views or policies. This slide here is to provide you with the standard or recommendations as it relates to establishing bioequivalence for LAIs within the FDA's product-specific guidances. One factor in the recommendations is whether the drug product can be safely given to healthy volunteers. If it can be, then typically a single-dose parallel study is recommended. Crossover studies are also possible, but often its uses are impeded by the long half-life of the product and thus the duration of the studies. If it can't be given to healthy volunteers, then patient studies would have to be performed. Depending on the PK properties of the product and the timing of subsequent doses, either a single-dose parallel study can be utilized or a multiple-dose steady state in parallel or crossover fashion. One of the barriers to generic development for LAI drug products is the conduction of these PK studies. While we recently have had some breakthroughs in this area with the first generic LAI approvals, many RLDs still don't have generic competition. The difficulty in conducting these studies lay in both the long study duration, oftentimes it would take more than a year, and the high variability and or large sample size that arises from parallel study design, the use of multiple sites, or just natural variability from intrinsic or extrinsic factors. When patients are involved, recruitment can be difficult, especially with the sensitivity of some of these populations, such as cancer patients or schizophrenic patients. And long study duration, regardless of subject type, can lead to additional issues such as higher dropout rates. Evidence, or MIE-based approaches, can be employed to create alternative bioequivalence approaches for LEIs. The first type of approach utilizing POP-PK modeling would still utilize the in vivo PK studies, but enhance their efficiency, for example, by exploring alternative study designs, which will be presented later in my presentation. Such an approach would be accompanied by a pre-specified model-based analysis plan of the in vivo study intended to be conducted. The second type of approach is through PBK or mechanistic modeling, where bioequivalence can be established based on in vitro characterization of the test and reference products in lieu of the in vivo PK study. In this approach, a virtual bioequivalence study executed off of the in vitro data would be utilized to simulate the results of the in vivo study. In all, we see increased interest in both types of approaches through pre-ANDA communications and through ANDA submissions. Regardless of the MIE approach, there are a number of considerations to have in mind when developing these approaches. Any method should be sensitive in detecting formulation difference. In other words, could the approach detect a bioequivalent product, and could the approach show a reasonable passing rate for products that are expected to be bioequivalent? Based on the intended regulatory use, sufficient model verification and validation should be provided in the submission. Lastly, the modeling analysis should be planned prior to seeing the study results, and communication with the agency is encouraged prior to submission, with special mention of the new MIE industry meeting pilot program. As I've already mentioned, POP-PK-based methods can be utilized to enhance the efficiency of the PK bioequivalence studies. Namely, alternative BE study designs could be used that either shorten the study duration and decrease the sample size. For shorter study duration, such designs could include in silico continuation to simulate steady state from non-steady state conditions, carryover adjustment to account for being unable to wash out in patient studies, switch over repeat designs, or alternative bioequivalence metrics associated with narrowed bioequivalence limits. For smaller sample size, such designs include incorporation of reference scaling, carryover adjustment here, but namely for the transition from a parallel study design to a crossover one, and other optimization of study design strategies. With these model integrated evidence opportunities in mind with POP-PK, I provide here two model master file use cases based on alternative study designs and or bioequivalence evaluation approaches. Both cases intend to shorten study duration with lower subject numbers compared to the conventional bioequivalence approach, which here is a two-way crossover steady-state study. The case on the left here demonstrates an in silico continuation approach where the PK study has the subjects receiving test and reference product prior to steady-state attainment. Instead, POP-PK modeling off the in vivo data collected is utilized to simulate steady state for both test and reference products in each individual. In essence, what the conventional study would have looked like if conducted in those same very subjects. The case on the right is a switch-over design where steady state is achieved for one treatment initially, and only a single subsequent dose of the other treatment is then provided to the subject. Here, the POP-PK model developed off of this in vivo study would be utilized to virtually simulate new bioequivalence studies in virtual subjects, such as a conventional steady-state crossover design, or even the single-dose crossover design with sufficient washout that wouldn't be possible in a real in vivo setting. I would like to emphasize that in both cases, the aim of the model master file is to establish the appropriate methodology and evaluation approach in a product-independent fashion that could be applied then to any product to assess bioequivalence in the same way that the pure statistical average bioequivalence tests function. Where the product does come into play and its specific nuances, such as the degree of accumulation and phenotypic differences in PK characteristics, is in the establishment of the POP-PK model from the in vivo data collected. In the end, the different degree of model uncertainty would influence the bioequivalence evaluation. One such example is that the higher degree of model uncertainty could lead to higher prediction or confidence intervals in the simulated study. Mechanistic model-based bioequivalence approach is based on the integration of key formulation attributes and physiology to predict PK. These models can be used to define safe spaces for in vitro metrics between test and reference drug products, guide selection of clinically relevant or in vivo predictive in vitro studies, and aid in the development of in vitro in vivo correlation or IVIVCs off of in vitro release data or dissolution data. For the mechanistic modeling model master file use case, let's say that particle size remains one of the critical quality attributes for a long-acting injectable suspension drug product. In this case, instead of conducting the recommended in vivo B study, an applicant may want to use a IVIVC model to demonstrate equivalent performance of their test formulation as compared to the reference listed drug. This is the opportunity to establish a mechanistic IVIVC as a model master file. The PBK model can be developed by accounting for the relevant local physiology, physicochemical properties of the active pharmaceutical ingredient, formulation critical quality attributes, that is particle size in this case, and in vitro experimental data such as in vitro release data. Once you have the model, the mechanistic IVIVC model can be established by validating the predictive ability of the model with in vivo PK data from slow, intermediate, and fast release formulations whose release rates are basically dependent on particle size. This IVIVC PBK model structure can be reused with an appropriate revalidation. For example, if other ANDA applicant wants to use this PBK model, they can revalidate the model with their own formulations. Throughout my presentation, I have demonstrated multiple applications of model integrated evidence that can be applied to support alternative bioequivalence strategies for LAI drug products. In each model integrated evidence application, the associated model master file will provide all the relevant implementation details, including verification and validation. With a model master file in hand for these approaches, the benefits would be in 1, saving industry time in redeveloping and submitting these methods each time they are to be utilized. 2, increasing industry confidence that such different approaches from the PSG may be permissible. And 3, saving the regulatory agency time but not having to reassess these methods across every single application of them. Lastly, I would like to thank Doctors Gong, Alam, Yun, Sakalozu, Fang, and Zhao for assistance in the preparation of the materials presented today. I thank you all for listening and look forward to continuing the discussion in the panel.
Speaker
I
Dr.
apologize that
Tonglei.
circumstances did not allow me to present live today, but I'm still glad to open this session on the applications of the model.
All right, good afternoon, everybody. It's really a great pleasure to be here. Thanks, Jim and the FDA organizer for the invitation. And so again, I'm actually pretty new to MMF, and they actually learned a lot this morning. So I really want to switch gear a little bit and just to introduce you what we have been working on over the last few years. And they hopefully that you can learn one or two things from my talk. And so I'm again a professor at Purdue University in the Department of Industrial and the Physical Pharmacy. And the department actually recently changed the name from industrial to the molecular pharmaceutics. And physical pharmacy is gone. And I can echo that sentiment is that when I actually was a student at Purdue, so actually I was exposed to formulation, manufacturing, all those courses. But then after I was a faculty member at the University of Kentucky for 10 years and came back to Purdue about 10 years ago as well. Then realized, again, the pharmaceutical industry or research-wise completely changed, right? And so now I really understand why, probably physical pharmacy is not there anymore. But So in a way, is I realized when industry saw the problems, right, they move on, and so why I first came back to Purdue is realizes we can make very good drug products, right, and we can study all the formulation, manufacturing process. We have beautiful, right, oral, tablets, capsules, but one thing is that we do not know how to perform inside the human body, right? So that was, my personal frustration as well as this motivation for this work. We started almost 10 years ago. So at that time, we call actually digital human. But at that time, we realized actually learning curve, at least for me, is huge. I was actually trained in chemistry and computer science or computation. And when I was a student, actually, I don't say I hated PK, but it's not my cup of tea, right? But then I realized, again, when we started this work, I fully embrace PK, right? And but again, at this point, I'm not say I'm really, for the regulatory sciences, I admire FDA folks. I know it's hard work, but nonetheless, so the motivation for this work is, if we can make very good drug products, right? Can we really make that connection between CQAs, right? All these properties, the loading, maybe dissolution rate, mechanical strength, with whole body performance, right? And I think traditionally, one way to make that connection is IVC, right? You still treat human body as black box, right? And for me, is maybe my view is negative here is. IVC for me is really, you really try to make connection between an orange to an apple, right? It's not a really mechanistic connection, right, between the CQAs and the in vivo performance. So work we started, we want to look at actually the GI tract, but realize that's actually not very easy, right? That's actually, digestive tract is probably most difficult system to simulate, right, in the human body. So we started actually inside of a GI tract, we're actually looking at the skin, right? And so idea is can we really predict with the physical chemical parameters, right, of the drug, and also again of the physiological conditions to use a bottom-up physics-based approach to simulate local transport and absorption kinetics, right? And couple that with whole body PK. So in this case, whole body, again, as you know, is whole body PK or PK modeling is top-down approach, right? You would assume compartments, all that. Even PBPK for me is still a top-down approach at organ levels, right? So in that case, if we can simulate local transport of absorption and also use a whole body PK based on, in this case, based on the IV injections, right? We will get all these parameters, right? Whole body parameters. Then maybe we're able to make the connection between the CQA, right, of local injected products with systemic exposure. So that was really the motivation at that time. But again, it took us a long time to achieve and to get all the results we wanted. So here really a nutshell of the model systems of the local simulation. I don't know if you can see it, but so in this case, we really consider the local vasculature and the lymphatic absorptions, and also consider the like dissolution kinetics and the local transport kinetics of a drug delivery system. So at the end, we are able to model the local transport kinetics and local absorption kinetics. So there are three physical fields, if you will. One is the deformation of skin, right? So we actually consider the skin as elastic, right? If you put pressure, it can bounce back, right? So that's actually a deformation of tissue. Second field is mass transport of the drug, right? And third one is interstitial fluid, right? Because the reason drug can move around is because interstitial fluid flow, right? So that's why they, we have the 3 different physical fields, and there are, there are arrows. They are actually physical parameters that can make that connections between these 3 different fields, right? So during the injection, as you would imagine that, we're going to push our skin, and the skin, because elastic, it actually, drive the interstitial fluid flow. That's going to help for local transport, right? But this one only capture the, the hydrodynamics at the local tissues. But behind that, there also, I didn't show here, actually, there is so-called source term for drug absorption, or maybe local metabolism as well. So actually last week, my student gave me some screenshots. Actually, we use console to do as a solver for partial differential equations. So I guess for this meeting, I thought maybe I should present some like model files. So here I actually screenshot of that model setup. As you can see, probably hard to see, but I can tell you is, there are also equations we can put in to the model, and also there are parameters, right? Parameters for the drug delivery systems, and also parameters for the local tissue as well, right? So this allow us to carry the simulation of local absorption and transportation. So this is really a busy slide. I apologize, and I get used to the Zoom meeting, so it's. But we already published a few papers on this model. So I really highlight 3 different studies we did. Actually, each one is actually a PhD thesis, right? So it's really hard for me to talk all of them in 5 minutes. But on the left, you can see actually we simulate subcutaneous injection of proteins, monoclonal antibodies. And you can see those, again, monoclonal antibodies can be delivered or can be taken up for a long time, right? Weeks, days, right? And so in these systems, what we consider actually is absorption is taken up by lymphatic systems, right? So we really need the local lymphatic parameters in order to consider the absorption kinetics component, right? One there actually is we compare the local concentration of albumin, right, with experimental determined values. And albumin seems to be the only literature data available that we can find. So you can see there's pretty close match. And also we simulated at end is a systemic exposure of albumin, right, and compare with experimental data. We also did a lot of other monoclonal antibodies as well. But one thing I want to point out is we have to do so-called sensitivity analysis. I think Andy mentioned that. Is the reason is there are a lot of parameters we do not know, right? Especially systemic pressure parameters like lymphatic, permeability, diffusivity, right? Partition coefficient. So we do not know, right? So that's why we could not do prediction, right? Instead, we do this sensitivity analysis. So in the middle, that's actually intramuscular injection of LNP, right? So we did actually experimental studies. We did LNP with mRNA, luciferase RA, and the 2 different injection sites. So we did animal studies. And then based on that, we run simulation, right? Simulation of LNP and compare with the experimental data, right? Once again, in this case, actually, in this model, we actually consider local cellular uptake and also local metabolism of LNP, right? And also there's a drug, so-called, again, mRNA release from the LNP as well. And based on that, we can couple that with whole body PK model. But once again, I need to point out is there are still a lot of parameters we do not know, right? For example, the membrane permeability of the LNP or MIA, mRNA. So that's why we eventually we do this sensitivity analysis to really identify the important parameters that determine the local transport and systemic exposures. The one on the far left is actually a subcutaneous injection of nanocrystals for long drug release. And so that system, I think the methoxate, And so we actually, in that one, we consider the nanoparticle or nanocrystal dissolution, right, locally, and couple that with whole body systemic exposure. So once again, there are many parameters we do not know, right? And so that's why we actually did a sensitivity analysis to identify importance of formulation parameters as well as the local physiological parameters. Right? So that's probably run a little bit faster, but so that's my summary slides. Again, this model physics simulation allow us to do this sensitivity analysis of important CQAs of formulation. And so simulation actually, once again, is physical based, is driven by physical by parameters, right? And there are two sets of parameters. Where one is related to the CQAs and the drug properties or formulation properties. Another one is physiological properties. And based on our experiences, I think there's tremendous need, right, to measure those local physiological and anatomic parameters. For example, what about the permeability through the membrane? Right? What about diffusivity in the tissue? Right? And for us, is because again, we were focusing on formulation, but that's again, I think our frustration, as well as I think the future need in this field is we need to fully understand how a drug product behave inside in the physiological condition, right? In order to carry this bottom up approach. Right? So currently we try to integrate this PBM population balance model of dissolution with the local fluid dynamic simulation and couple that with whole body PK. Right? For the population balance, I just briefly put an equation right there. Is in this case we can consider the particle size distribution. Right? So that L, that's again related to particle size. X is the position, T is time, right? So this again, you can see the particle in the tissue right there is driven by diffusivity, as well as the movement of fluid, right, convection. Right? And then on the right here, that actually is so-called South term. That's again related to the dissolution, and maybe if we have a super saturated solutions, we may have recrystallization as well. Right? So for that one is partial differential equations. Right now we try to again use MATLAB actually solve the partial differential equations and couple that with CFD simulation by COMSOL. And for PK actually we are using MATLAB as well to do all these calculations. Yeah. So that's current work. But I think early, my next speaker, Andy, actually going to talk, hopefully, sensitivity analysis. Based on our experiences, again, we call this modeling and simulation, right? We don't call this prediction, right? We cannot predict, right? Because again, there are so many parameters we do not know, right? So far, I think the state of art of S&M simulation modeling gave us a platform, right, to really explore the design space, right, of maybe in the drug products. But on the other hand, this simulation can take a long time, right? Each simulation, as you can imagine, in the partial differential equations, it for us, take days to solve those equations to really achieve the converged results, right? So with that, it's really hard for us to do a global sensitivity analysis, right? For example, CQA, right? We may have 10 design parameters, right? For each parameter, if we only sample 10 times, right? So that will be 10 to the power of 10, right? And if you one simulation do a week, that's that's cannot solve that things, right? So what we are thinking, and also actually talk, just interacting with engineering faculty at Purdue, we have fabulous faculty, engineering faculty. For those people, they want engineering, they want to simulate everything, right? So one of the things we learn is, can we combine the simulation with machine learning, right? With machine learning. So one of the model is, you probably heard, it's called the surrogate model or hybrid model. So we can use simulation to represent the data points, right? And may project that design space to a lower latent space and they run simulation models, right? That's why, I said when I was a student, I'm not a big fan of PK, right? But now I fully embrace, again, the PK and the Bayesian model as well. So in a way, we may be able to combine the physics informed simulation with Bayesian, right, based modeling. And I think as a scientist, right, I was trained in chemistry. As scientists, we care about the mean, right? We care about average value. But again, interacting with engineering folks, what they care or what they concern most is uncertainty, right? It's distribution, right? So it's nice to have one value. That's actually physics-based model is if we set up the parameters, you always get the same value again and again, right? You don't get a distribution, right? So hopefully, again, it's by combining the physics-based simulation with data, right? Data science, we should be able to, get this distribution, right? And that's, again, uncertainty quantification. And I think for industry, for FDA is, again, that's actually very valuable, right? Risk assessment and uncertainty quantification. Right? So I know my time is up. Last point, I actually was thinking about this, I'm truly support, FDA effort in the MMF. But on the other hand, I think there's also a need in education, right? And I think is, our students get used to, and still I think it's pretty heavy, it's data, data-centric research, right? Experimental heavy research. So how can we really, make that, change the curriculum so that we may have a data, driven research, right? So right now we, at least in my department, we don't have much emphasis in modeling and simulation, or even data science, right? I think that's need to be done, and hopefully FDA will play a major role in driving that. Okay, thank you all.
I'd like to welcome Dr. Andy Hooker.
So I'm going to talk about model-based bioequivalence methods that we can use to determine if long-acting injectable products have bioequivalence or not, and potentially how we can use that, incorporate that into an MMF. And of course, it's not just me doing this work. So I've listed at least some of the people other than me that have been working on this. So we've been working on a number of different projects in how we can use model integrated approaches to evaluate bioequivalence. And we started off looking at what happens with sparse data. And then we went forward and looked at long-acting injectables. And then right now we're looking at highly variable and long half-life drugs. So we're sort of investigating a lot of different potential areas where we think models can be more useful than standard methods. And why are we doing this? Well, we're doing this because sort of the standard, if you think about the standard bioequivalence for clinical bioequivalence studies, you might do a two-way crossover study where you're then computing these summary PK metrics based on non-compartmental analysis. And these non-compartmental analyses approaches tend to have problems if you have sparse data, if you have highly variable data, or maybe if you can't achieve a steady state, or you can't run these crossover studies. And that's something that Andrew mentioned in the first talk in this session, where he was talking about the problem of running these types of studies in patients where they have to be on treatment. So you might be running a steady state crossover study. And if you're in long-acting injectable drugs, then that steady state to steady state type of crossover study can take a very long time. And so we generally think that models, if you have a model that relatively well describes your data, then they typically will be more powerful, so you should be able to reduce study size. You can also do things like not run you can extract information from studies, from designs that aren't crossover, or they don't have, you haven't taken all of the washout that you might need for a standard analysis. So our method that we've developed, it essentially goes like this. We have some sort of data. That data doesn't necessarily have to be a crossover study with washout. It can be some design like Andrew was talking about. That could be some modified design. Then you have some models that you think should describe that data. Maybe one model, maybe a couple of them, or multiple models. You need to then adjust those models, adding in treatment effects, potentially according to dependent on the design. You may have to add in sequence and period effects. You want to add in inter-occasion variability, and all of this needs to be added onto the parameters that should be changing between two formulations of the same compound. So you're looking at the absorption properties of the drug. Once you fit the model to the data, you also need to estimate how certain you are about those model parameter estimates. And so we spend a lot of time worrying about uncertainty of our parameter estimates and the distribution of that uncertainty. And once you have your model, your parameters, and the distribution of the uncertainty of the model parameters, then you can do simulations. For example, simulating out a gold standard two-period crossover study, or a four-period crossover study if you're looking at highly variable drugs, and figure out what, in those idealized types of designs, what you would expect the geometric mean ratios of Cmax and AUC to be. So then you can do simulation and draw conclusions based on what your model is telling you. And these methods in general have shown to control type 1 error. On the left side of this plot, you can see the overall type 1 error for both the model integrated approaches in red, and in blue, it's the standard NCA approaches. So you can control type 1 error, and in general, you have higher power, and this was for the sparse data example. So we could see that with sparser data and more uncertain, so more variable data, then you would have more power with these model-based approaches compared to standard NCA-based approaches. When you're thinking about long-acting injectables, then one of the problems is having to run steady-state crossover studies. And so this is just an example where you can imagine that patients are on a reference product, and then you want to test the test product. So you have them on the reference product, you have to make sure they're at steady state, and then you switch to the test product, and then you have to wait a long time before they get back to steady state, and then you can do tests based on those two periods of the last steady state periods for each compound. Our idea was that with a model-based approach, you can sort of subtract the amount you expect from the reference product in the first and second periods of the test periods. So then you could run, for example, a switch study where you have people on steady state of a reference product. You give them 1 month, or so one dose of a test product, and extract the information you need in order to simulate out what a steady state crossover study would look like. And in this case, we used as an example paliperidone 1 month. So we're looking at 1 month doses of the drug. So instead of having something like a 36-week study, you might have something like a 16-week study, or even less. Now, why would this be useful? Well, one thing is that actually determining steady state is sort of problematic, I think, especially if you have something that's variable, lots of residual variability, lots of between subject variability or within subject variability. Then you typically, the way you identify steady state is you're looking at trough concentrations in 3 periods, and if you see that there's a straight line or a flat line, then you say you're at steady state. And that's with lots of variability. You can very easily identify that you're at steady state when you're not. And so we were doing some simulations where you could see just by changing the absorption rate in the test compound that it might take twice as long to reach steady state than what you might think from a standard study, and then you have a high probability of identifying that you think you're at steady state when you're not using these methods. So the model-based approach and model-integrated approaches don't require that you that you're at steady state for the test compound. These approaches where you look at say the first period after switching to the test compound, they can actually be more sensitive. So you might be able to see differences with fewer individuals for absorption rate. Absorption extent, it tends to be less sensitive. So you might need more individuals in order to identify whether there's a difference or not in differences in bioavailability. And so if we simulate out some studies where we assume that we have a bioequivalent compound, but there's some difference in the two in terms of absorption extent, then you could see that with these switch studies that you could really shorten the study quite a bit, but you might need, at the worst case, 6 times more individuals. In the best case, you'd need fewer individuals if it was just absorption rate that was different between the compounds. You could then start thinking about, okay, can we expand this study? Can we reduce sample size even more by, say, looking at the last 2 periods before switching to the test compound and then looking at the first period after a switch? So then you have a 3-month study per individual. And you could see then a reduction of 10% of the total sample size needed for identifying whether there was bioequivalence or not. You could then also sort of think more and say, okay, well, let's look at the first the 2 periods before switching and 2 periods after, and of course you get more and more information in these types of studies. So we could control type 1 error, we could shorten the studies significantly, but the question is sort of how do you do this in reality, right? We have a requirement that you have to have models or a set of models, and it's either models from a previous reference product, or potentially if you don't have any models, you may have to have some sort of model building procedure, and if you want to maintain type 1 error, then that model building procedure better be predefined and done in a special way. Then we have to adjust these models. So even if you do have a reference model, we need to add in terms in the absorption portions of the model in order to identify the differences between the test and reference compounds. So that's important, is adding in these extra terms. There's another problem, and that's if you have a reference model from a reference compound. Oftentimes, it's not been built on data that's only been a bioequivalence study. So these models have been developed based on a number of studies, and so you end up with a model that may not be identifiable in just a bioequivalence study with that design. So it could be that you then need to figure out how to reduce the model in order to make all the parameters identifiable. And that then leads to potentially reducing it in a number of different ways, potentially different models that you need to use in some sort of averaging setting. Lastly, then you need to be qualifying the models, showing that the models can actually fit the data. So I'll show some pictures there. If we're thinking of long-acting injectables, there's generally at least of the 33 products that were available in 2021, there was 17 products that had published models and roughly 2 published models per substance. So then we have that problem of choosing which model makes sense or potentially using both and then averaging across them. And again, those models oftentimes are based on studies that are more complex than a standard bioequivalent study, so you need to reduce the model in some way. So these model averaging approaches need to be used, and actually that result I showed you where you needed 6 times more individuals in this worst case scenario for a switch study, that was done using a model averaging approach where we assumed a number of different models for a paliperidone situation. So I think it's important then to think about model qualification, and so at the minimum, then your model should be identifiable when you're estimating parameters. So you need to make sure that the design allows you to estimate parameters of interest, which you can test with the optimal design software, for example. You also need to do some sort of simulations from the model to see if you can recreate the types of data that you see. So in this case, I've just shown one example where I've simulated data with the model and then computed non-compartmental analyses of AUCs and Cmaxes and computed the means here and the variances and compared it to the true data set, or a simulated true data set. So in this case then, how do we sort of try and allow people to use these methods and allow people to use the models? So we have a package called BEMod written in R, which sort of works the full workflow of starting from a model, estimating data, estimating parameters, estimating the uncertainty, then doing a simulation of the gold standard study and calculating these summary statistics. You can then think, okay, well, if we have a model like a paliperidone model or a set of reduced models, then we can add those into a database and then apply that to this BEMod package. So we have a package called BEMod LAI, where we've taken these paliperidone models that we think are best and to use in a model averaging situation. So at least from an academic's point of view, I always sort of think that it's quite useful to have things open source. So this BEMod package will be coming relatively soon, hopefully in the next month or two, to CRAN for the packages where the repository for R packages. We then have these other tools that we also need for this approach that are all also open source, so you can see what's happening. You have version control, and you can see what sort of changes have occurred. All right, so then how can we think about a model master file in these situations? And I think if we think about the package itself, this BEMod R package, that's sort of describing the workflow, and it's implementing that workflow. And that, I think, is anyway one thing that these MMFs could be useful for is as a qualification tool. And I think Flora is going to talk a little bit about the qualification tools at EMA. I got a preview of her slides the other day, and I think there was not very many qualifications in the European Union. 0 this year, is that? Yeah, okay. So at least sort of figuring out how to get people to try and qualify their methods with the MMF is perhaps a challenge. But I think it's useful, at least from an academic point of view. I would be interested in trying to get my method qualified so that people would think, oh, it looks like it could be a reasonable approach, potentially. In just these approaches, we had specific models that are needed for a specific drug and a specific design. So all of these so it's very specific to the situation. So I'm not entirely sure how relevant it is to have the specific PK model in a model master file, because it really is specific to the drug and the design that you're interested in looking at. But we did hear this morning, Dr. Leyenberger, he talked about the idea that maybe you have 1,000 generic drugs, generic companies submitting for the same compound or the same generic. Maybe in that situation, then having some sort of MMF with the drug, with the model for just that situation could be useful. So I've sort of been struggling with trying to figure out how you can get companies to create these MMFs and then share them across organizations. Because I guess that's where, at least for the FDA, that's really where you get the benefit. They don't have to keep on reviewing the same thing over and over. And I think I see it. I think, you know, if you're the first generic person to come in with a model master file, then you can sort of sell it to the next person. So I think after listening to our talks here, I think I see the business case why people might want to do that. But I would like to sort of put out at least a little shout out for public model sharing, because I think there's a benefit of having a completely open model that you can actually see what's happening, and you can then see how it was used for its specific use case, and you can adjust it for the use case that you specifically have. We've been talking about these sort of model packets that you can plug in, and I think oftentimes you're going to need to adjust those models in some way, or maybe you're interested in the correlations between the PK and the PD. You need to have more control than just these plug and play types of models, potentially. But I think the public model sharing can be useful with standardization, improving new developments, and explicitly validating and verifying the models that are being used. So I think the big benefit I see for an MMF is, for these types of population PK models, is the methods and qualifying or verifying that the methods are a reasonable approach. And then specifically sharing the drug models for the specific design may be interesting, but I think it depends on the scale yeah, I have a harder time. I can't decide whether it'd be useful or not. We can discuss it in the afternoon. Thanks very much.
Thank you, Dr. Hooker. Our next speaker is Dr. Daniela Silva.
Okay, thank you. So yeah, I'll be talking about the application, MMF application in the context of LAIs. So here's a little outline of what I'll be covering today. So I'll start with a case study to serve as a background to the MMF concept, and then go over a brief summary of the findings, and then understand how could MMF could be applied using that case study as a background. So basically, this was a subcutaneous injection of a low solubility compound, suspensions in rabbit. And we had 5 different formulations, one being an RLD and 4 Q1, Q2 formulations. And they differed in particle size and dissolution as well. We developed a baseline PBPK model, and that model was calibrated against IV CP time profile data. So what we wanted to see is that can the PBPK model then link the formulation parameters to in vivo exposure for these formulations? So before I go, I cover about like a little bit of the model development itself. I guess I'll cover a few different principles when we're talking about LAIs. So when we think about the GI tract, we kind of assume that the particles are well separated, right? And that the total surface area of each particle would play a role in the solution. But when we think about the subcutaneous or intramuscular space, we have a much more restricted tissue environment. And this can cause packing or aggregation to occur. So that packing or aggregation will then affect the effective dissolution surface area. And the second thing that we need to think about as well is that In the GI tract, you might have like a reasonably well-stirred environment, and that keeps the diffusion layer thickness kind of small. Whereas in also like a subcutaneous or intramuscular space, you have a more static environment, and that can affect the dissolution layer thickness, which may be significantly higher for in these type of injections. So the first, now I'll go over the model development and results. So here, the line is the simulated profile, and the squares, the empty squares, are the observed data. And we see that, you know, the first approach that we took was just to take the experimental particle size distribution and just a standard unstirred water layer, or that diffusion layer thickness. And we see that we might have been kind of like capturing the AUC, but obviously we were not capturing the shape of the profile well at all. So the first thing that we did was to increase the diffusion layer thickness, and we saw that that had a significant impact on the profile shape. But as you can see in the middle, the profile is still not being well captured. So then what we did was to scale the measured particle size, and use kind of like a scaling factor for that PSD, and we saw that using that approach further improves the shape of the profile, especially for the end tail of the profile, so the terminal phase. But we can see that the Cmax was still being underpredicted. So after literature research, we see that we saw that the effective depot volume can increase, have like a temporal increase due to inflammation. So when you inject something, inflammation can occur, and that means edema. So what that means is that the effective depot volume, or the volume where the injected particles will have to dissolve and then partition to systemic circulation, can be higher. And as the inflammation subsides, that volume kind of goes back to the baseline volume. So when we included that inflammation in the model, we were then able to very well predict the shape of the profile as well. So then the next step was to apply the exact same approach to the other formulations, since, as I said at the very beginning, that we had 5 different formulations. So here I'm kind of like showing a graph with all all the formulations, all 5 different formulations, and we see that we used the same scaling same scaling for particle size distribution across all formulations. And then we see that the model was able to distinguish the differences in exposures for these Q1, Q2 formulations. And as I said before, they had different particle sizes. And the scaling of the particle size distribution suggests that aggregation happens slowly and over time. And lastly, we see that a combination of these effects, so slower diffusion, aggregation, and inflammation, was required to correctly describe the shape of the profile. So taking this as kind of like a background, we see that when you have obviously a verified and validated model, and then you have that mechanistic PBPK model that describes the mechanisms impacting the in vivo dissolution, physiological parameters, and absorption processes of a compound, you could have a few of kind of like quantitative approaches in terms of applications of an MMF. And this, I guess, is also the same as was covered in the morning about oral drug products. So if we're talking about the same API, the model could be used to describe the pharmacokinetics, right? So once the compound's been absorbed, the pharmacokinetics will be the same, so that could be applied. And the post-dissolution processes as well. So if you see at the bottom diagram, if we're talking about a suspension, that particle will dissolve, and then the dissolved particle will partition between the tissue environment to the systemic circulation. So that partitioning process can also be translatable, since it's kind of like a post-dissolution process. And physiological aspects as well, right? And that's, I guess, also kind of related to the platform. So physiological aspects such as the blood flow, tissue compositions, and things like that, they are translatable across different projects or different drug products. Now, I guess potential further applications in a more qualitative approach, or kind of like lessons learned, I guess, from the MMF or from the modeling exercise, I guess I'll start from the bottom in terms of the inclusion of inflammation or inclusion of particle aggregation. So if your product is also a suspension, aggregation might occur, but also it wouldn't be like, if you needed to scale the particle size, you need to derive your own scaling. And in terms of inflammation, we need to take into consideration that it's not just, suspensions that might trigger inflammation. The physical chemical properties of the API itself might be enough to trigger inflammation. So that's more of a harder, I guess, parameter to kind of translate between MMFs. And I guess going to the upper part of the qualitative part, and I think Andrew covered this as well about the mechanistic IVIVC. So the same auto structure could be used to obtain that in vivo dissolution, but obviously you would need to develop your own IVIVC. And you could also qualitative inform the model development for similar types of formulations. And with that, I'd like to thank you all for listening.
Thank you, Daniela. I'd like to welcome all the speakers and panelists to the front and join the panel discussion. So in addition to the speakers, we have the additional panelists, Dr. Khondoker, Dr. Marie Ducharme, and Dr. Rebecca Moody. So I'd like to open the floor to the audience, see if you have questions.
Yeah, it's not a question, it's actually an invitation. Andy, you should come with your model for qualification, actually. I think, I think this will be increase also the trust into this kind of platform for being used not only by industry, but also by academic group. The only issue that I can anticipate is the validation data set, because it will be so open that we need to agree on the space you need to validate or to qualify your platform for. But at least an ITF meeting, why not, to discuss all these components and even to agree on a context of use where we can delimitate already that you do have enough out there to claim that the platform can be used for that purpose. We need someone to start. Thanks.
Okay, we'll meet, I'll be there.
I'll just talk loud. Still? Yeah, there we go. My name is Jim Bosley. I'm with Nova Discovery, and I've been doing QSP modeling for the past 25 years. So a lot of this is, with the pharmacometrics focus, has been how applicable is this? I was really interested in your, Dr. De Silva's example of developing the model and adding physiological insights guided the development. Dr. Lukakova and I were talking, and she made the comment that I interpreted at least as for a physiological model, large-scale QSP model, the knowledge model that you use to build the model, all the PDFs, is essentially the model master file. I don't know if you agree with that, Dr. De Silva, or Dr. Silva.
Yeah, essentially, yeah, like the approach and then kind of like the documentation if you need to do so at the end, kind of like building gathering together everything that you did to build that model would be essentially an MMF.
I guess the advantage of having an MMF would be that it's not only the people who have been building the model that are evaluating it and saying that it's good, but it's also there's sort of an independent review of some sort. So I agree that it would probably be, that would be the contents of the MMF might come from what's been done in building the model, but then somebody outside of somebody with less less stake in the game is there to review and say, yeah, we also think it looks reasonable.
This is a question for Andrew Booker and Tonglei, is that how you pronounce your first name? Yeah. It wasn't clear to me in either one of your talks what statistical framework you employ after all of the modeling and parameter estimation that leads you to a yes or no bioequivalence. Are we talking about toast? Or are we talking about some other heuristic?
I can answer first. Yeah, I was using toast, but I'm also aware that there's other frameworks like the one that Franz Montre has been involved in developing, where you're looking at these chi-square distributions instead of toast, which I think looks like a positive alternative that can make your model-based approaches even more powerful.
So if I can follow up on the question, why choose toast? Why are you fitting to the normal distribution when data and often parameter estimates deviate significantly from the normal distribution?
So we're not assuming a normal distribution. We have our model, and we're generating a confidence interval based on our model parameter distribution, the between subject variability, residual variability distribution, and then the uncertainty of all of those parameters will generate a confidence interval that's not normal. It's a non-parametric distribution, but then we compare it to the limits that are expected for a toast. So we're not assuming that our distribution of Cmaxes and AUCs are normally distributed. We're just trying to fall within the limits of the standard bioequivalence test.
But they're log normal distributed, right? I mean, you're using non-mem?
Not necessarily.
Okay.
So you certainly could make the assumption that you have a log normal distribution. If you have log normally distributed parameters, then most likely your distribution of AUC and Cmax look pretty log normal. But it's not a requirement for the method. You could have whatever types of between subject variability distributions that you like.
Maybe I wanted to just see if there are any online questions, Yuqing. And maybe Andrew, if you wanted to chime in.
So we received 2 online questions.
Yep. Before online question, can I have 2 questions here?
Go ahead.
So I don't know, I just want to make a comment whether trying to make Andy particularly happy, since we haven't put that fact into the operational fact of the sheet yet on model master file. For model master file, the content can be published. Actually, we encourage you to publish that. It's not a patent. You know, for proprietary information, people have the option to keep it as a trade secret. Then, however, if anyone outside have found the same thing, or you yourself are willing to publish it, people are more than welcome to do that. Actually, if you put it into the publication and have gone through peer review and also comment from the public, that only gain FDA confidence. Okay, this model is trustworthy. You can use it. The existence of the master file can offer more like a record keeping or like a media to cut the communication. So we have communicated for this model to this stage, and here's what we have agreed. Now we have record, there's agreement. You know, there's no means that model master file that's not the intention to use the model master file to stifle competition, public scrutiny on the model development. That I just want to make that important statement.
Yeah, that's a point well taken. And of course, it's as an academic, then there's the one is driven by different factors than somebody working at a company. So oftentimes that's very important to be able to publish things. So I think that's a great alternative.
Hello, it's Amin Rostami, University of Manchester, and also Tara. Just two comments. I think, and Andy already mentioned, on sensitivity in one of the questions, but because Dr. Tonglei also, I think, emphasized on sensitivity, I wanted, to comment that we have to have a little bit of sensibility with regard to applying sensitivity analysis. Because I think most of the time, particularly my colleagues who are coming from engineering background, maybe they are actually missing the fact that in the biology, we have got lots of correlations between different parameters. So when you are looking at, for instance, let's say long-acting injectable and your model, for instance, is sensitive to albumin in the place, but at the same time lymph. And both of them, they are having, same direction of effect. By changing one parameter, saying lymph, and then say, oh, it is sensitive to this degree, that degree, already makes the assumption that there is no correlation between lymph and albumin. If it happens that lymph and albumin, they are inversely correlated, no matter how much you are changing the lymph, because the other one is going to compensate, it's not going to have an effect. So when we are building these models, we have to make sure that we are considering all the correlations that they are known. And unfortunately, my colleagues, as I said, they are coming from engineering background. Most of the time, they are actually ignoring when they are available, or they are assuming that they are not available, which is not true. That's one comment. And global sensitivity analysis doesn't answer that because it's basically doing sensitivity in different values of different parameters. It is not looking, it's looking at their interaction, not correlations. The second comment, is regarding, this discussion on availability of the MMF, for everybody to look at. Why we are making a distinction between MMF and, a drug master file or any other submission that goes to FDA or any other regulators? By saying that, we want all to have MMF, sort of for approval, we are saying our colleagues in the regulatory, when they are deciding something is approved as MMF, they are incompetent with regard to that decision. I think this is not right, because those people who are sitting and making that decision, they are making decision on the basis of good judgment and scientific judgment. The fact that it's not available for me as Amin Rostami doesn't matter, because they are qualified to make that decision.
Okay.
Well, maybe I want to comment. I'm not sure. I mean, are you responding to the comments from Liang? Because I think Liang was not forcing that it would be public. It's just that if you wanted to publish it, then you can publish it. Okay, but if you don't want to publish it, then you don't publish it.
No, I am commenting with regard, sort of the models that in the form of MFM, when we want to have trust in them, should be available to everybody. That is not necessarily the case for anything else. Why we are making an exception? The only people who are supposed to be making a judgment with regard to the MMF acceptability are those people who are actually receiving the submission. If they see that this is qualified, everything is done, etc., that's their decision. I'm not going to, to say, Flora is not, sort of qualified, to decide whether this is acceptable or not, because it's not public. But of course, Flora can say, these elements that you have put in the model, I want to see actually all the information that they are either in the public or, submitted to me that I can judge. I am not going to accept if they are not transparent. That is their right, but to assume that these need to be available to everybody, that's basically is making an exception compared to anything else that happens with other aspects of drug submission.
I think you can have both ways. Some may not be available to everybody, and then others you may publish and it's available to everybody. There's an advantage when it's available to everybody because, there's more eyes looking at it and you never know. But, yeah, I think that was your point, which was excellent.
Does anyone have one? Yeah.
Maybe I can comment. If you continue talking, I think.
I can comment something on your sensitivity analysis. You bring a very good point, actually, the sensitivity analysis. Often we think that the sensitivity analysis should be done on the formulation attributes to identify that what are the sequelae that will be different between reference and test drug product. But sensitivity analysis can be also done for the physiological parameters when you are developing the model, right? But one thing I want to also mention here that sensitivity analysis may not be very useful if your model is wrong at first place. So your model is, if it is suitable for detecting the formulation difference, you may conduct the sensitivity analysis to understand more that what are the other parameters, what are the other CQA that can influence the systemic or local tissue exposure, right? So we need to be very little bit careful to applying the sensitivity analysis in a general framework. Yeah, right? Yeah.
Are there any other questions?
Yeah, and probably follow up with all the questions. I think from my personal view, I think there may be 3 layers of complexity. One is model. Model is really based on, as I think Carl mentioned, the Newton, right? There's model, and model can be published, right? Can be shared. There's second level complexity, I think, is parameters, right? So parameters may be related to drug product, CQAs, or even the biological different conditions. And third one will be, I think, the regulatory aspect. Right? And so I don't know. For me, I'm working on the science. I'm open source and share, publish. But for me, I don't have access to the parameters of different products. And but I can still develop a model, can still share the model and test the model. So that's again, I don't know if you mentioned, but you also asked for the normal distribution. I think one way to address that, actually, over the last few years, we're doing the AI, but most of AI model is really based on Gaussian distribution. And you can have multiple multivariate Gaussian distribution, which can approximate any kind of complex functions. Right? And the beauty of Gaussian is the derivative is the same. It's the same thing. So I think that's I talked to yesterday. Have conversation is the Gaussian function, right? Is so important. Yeah.
Yeah, thank you. I actually have a question for Dr. Lee. So it is a technical question. In your last slide, you had the CFD, and I thought it's something significantly different to the other models that we have seen today. Could you, if you could elaborate more on the application of the CFD for the LAI? Because you don't have any momentum transfer, no fluid flow, no Navier-Stokes. So what's the advantage of solving CFD for those long-acting?
So in our system, again, this good point is actually the convection is pretty trivial. Mostly, actually, I didn't show that actually drug, the formulation mostly concentrated right around that injection region. So during the injection process, the elasticity actually play a major role to drive that convection process. But after that, after maybe a couple hours, then the process is mainly driven by diffusion. But that's models, right? People said all models are wrong, but some would be useful. So we, always make assumptions. So maybe in some cases, for example, disease state, I think, mentioned that inflammation. So how about the local inflammation can drive, convection? So that, again, can be simulated by CFD, I guess. Yeah.
I'm more confused now. If we don't have any convection, why do we need CFD? You said this is diffusion driven, but we need CFD. So these two are.
Well, there is actually convection. There is interstitial fluid flow, right? So in our case, actually, the tissue is considered homogeneous, right? So because when you consider homogeneous, at each point, the net flow is 0, right? But doesn't mean there's no flow. There's still convection. And if system is not homogeneous, for example, around the injection site, there's still convection. Yeah? Because again, the concentration is going to drive that. Yeah.
Okay, thank you.
Sure.
I think Andrew online had a comment on the previous question. Andrew, I don't know if you want to still comment. Yeah.
Well, first off, can you hear me?
Yes, we can.
Yeah, I think we already kind of moved on to a completely different issue. But when we're having that conversation about publication and sharing, and I think we're still seeking more of the mechanistic modeling perspective. But if I go back to like the POP-PK methodology one, on whether what's proprietary or what's keeping hidden or what's not published. But I think if you're going to do this novel B approach and you're going to get approved, that information is going to be available, likely through Freedom of Information Request Acts. Like you're able to go to any ANDA, see how it's able to be approved. So people think if they're going to be able to they have this secret super method they're going to get bioequivalence and keep that from everyone else. No, like that information will become, I imagine this is my guess, like will become available.
And hi, this is Stefan again from University of Macau. And it is about the question for the long-acting injectable formulation. Just now, the several speakers talk about the different QSP models for these long-acting injectable products. And my question is, for the long-acting injectable formulation, actually we have different types, right? For example, the PLGA-based, the microspheres, and also the gels, and also the nano suspension. So when we build the PPK model or QSP model, it's possible for a general model for all these types of the long-acting formulations, or we have to build it individually, especially when we try to link the formulation parameter with the QSP models. Thanks. Yeah.
When you're talking about different types of formulation, like I said at the beginning, and others have said as well, if you're administering the same API, then you could use, the post-dissolution process or the disposition model for that particular drug would be translatable between drug products. But if you're talking about different mechanisms of release, for example, then you would need to parameterize that particular product. So you couldn't really translate one MMF that's like a suspension to a PLGA system, for example. They would need to for sure have, like, that part of the model would have to develop, if it's not developed yet, be developed and validated, and validated to be applicable. I don't know if others have different opinions or want to add anything else.
No, I mean, I completely agree. But I was just going to say that, in terms of formulation variants and things like that for LAIs, like, we still need a better understanding of what's happening, what's impacting solubilization at the site of injection, and what's causing dissolution. And then we need better in vitro tools so that we can, put these in PBPK models and things like that.
Maybe can I just comment on this thing?
Yeah, go for it.
I think absolutely what you just said. I know we talk a lot about mechanistic modeling and all of that. We're not there quite in terms of LAIs. And what you just said, I think that's why the in vitro tools, even in terms of, you mentioned imaging and the depot volume and all of that, those fundamental inputs will then help in getting to that mechanistic modeling. Because you had, go ahead. Just why I had to say that.
Maybe I can add some thought here as well.
I think I just wanted to make a comment on the open source one. So 99.9% people here can drive a car. They can do a decent job. But they don't want their engines open to tune up every morning or check it's working. They have to trust the mechanic or the provider that, engine is safe. When something goes wrong, they can go to the mechanic to get it fixed. If you give me your R code as an open, I would be more worried that I would mess it up. I want it to be foolproof, not open, to be able to use it. Maybe somebody who is very, very good in R can check that code, make sure everything is good. But when you give it to large majority of the people, it should really do what it is supposed to do, not necessarily open in terms of every equation. But that's, again, my view.
I think we can argue all day about whether we should have open source or closed source software and systems. You can also argue that Microsoft had Excel for 30 years and they couldn't compute a standard deviation correctly for 20 of those or something. It was some very easy function that they had never fixed. And of course, it's closed source and you can validate it, but it's wrong. So if you have an open source system and somebody finds an error, you can easily figure out what's going wrong and fix that, as opposed to hoping that the people who actually have control of the code can fix it. But I agree there's arguments both ways.
Yeah, and the discussion is really about PBPK, because for PubPK, it is, everybody, it's open source, you can find it. Things are published and more people can review and all of that, but it's less complicated. One thing I want to say about the model master file for long-acting injectables is that I see it as more than the model. It's similar to what Dr. Babiskin presented. If you do, for example, the in silico continuation approach, it's not just the model. After that, you have to think of an innovative study design. You have to show that this innovative study design controls the alpha error. It's like a lot and a lot and a lot of work that can take years. So I think for long-acting injectable, model master files should also have included all of the proof that an innovative study design controls the alpha error, etc., more than just the model. With PubPK, obviously, here that I'm talking about.
Maybe we have about 10 minutes left, so I wanted to get an opportunity at least to go to a few online questions maybe, and then come back to the room. Is that what you want to do? Yeah. And I think, again, listening to this conversation, right, there's an overarching MMF, whether it's for oral or long-acting or any modality, open source, closed source. Some of these, I guess, the debate will be during the next session, I think, coming back to probably a bit more specific about long-acting. One of the questions online, has modeling simulation been deemed acceptable for generic long-acting injectables? And are there specific concerns and challenges that we should be mindful of when it applies to long-acting injectables? Specific concerns or challenges. So we heard, and I think, again, reflecting, we heard very well-established concepts, Tycho, others, we're talking about even templates. Here in this session, it's more of concepts, and then not only modeling concepts, but as you just pointed out, it's how to take innovative study designs and so on. So maybe the panel can comment on specific aspects as it relates to long-acting. That's the question.
Hello, can you hear me?
Yes, Andrew, please go
Yeah,
ahead.
I'll take a crack at this one. I think it's directed to the FDA. As I said in my talk, we just had the first generic approvals for LAI. And to my knowledge, there was no modeling involved in the approval. They were just following bioequivalence as we stated within the PSG. If the firms use modeling simulation for their product development, I'm not aware of it. So it's still a developing field. If there's experience with it, so I'm talking from the generic perspective, but there's experience with model simulation on a new drug, I'm not aware. So if someone else can chime in if they know that. But then again, from our own internal practices and setting the standards for long-acting injectables, we use modeling simulations, for instance, for establishing the partial AUCs that you'll see in some of those recommendations and other things. But that's really where the current status is. This is where we haven't had, until recently, we haven't had generic approvals. And so this is an area where we really need that innovation in to really, so you start to see more applications, more approvals.
Anyone else wants to comment?
Well, I can comment a little bit. I mean, we've been developing a method on how to establish B with model-based approaches for long-acting injectables, and we've had meetings with FDA that has been very successful for multiple products. But it's just that it's a big step for generic industry. I don't remember who was talking about it this morning. I don't know if it was you, Carl, or someone else, but between the standard and the new approaches. And when there's even though we we went to that step with FDA, and FDA said, okay, it looks good, you can go ahead. It's still a new thing for the generic companies, and they're kind of, I don't know, they're stopping their, they don't know if they should take the risk. And they prefer to stay with normal standard design. So for example, that's why for long-acting injectables, they prefer to do the 300 patients parallel design, because then it's a standard approach and they feel that it's less risk.
Maybe we'll take one more online.
I would also add that
Go ahead.
some of the things that can go wrong in the traditional studies could also go wrong in these novel approaches. But yeah, someone has to just, it's an innovative approach. Someone has to be willing to take that risk to push it forward. There's only so much advice and recommendations that we can give. We're not conducting the study. We're not doing the power calculation. Eventually it's going to fall on the firm to try.
Yes.
Okay, go ahead. Yeah, please go ahead.
So maybe we should abbreviate MMF with a lowercase m and bold uppercase F, MF. I want to bring back again the discussion on the master file part. Let us say that somebody submitted a small m, big F to the FDA, and the FDA reviewers accepted that model. Now I'm going to play generic. So would I take my, let's say this is the LAA session, a new LAA test? How would I run or access that model? Is this going to be something like a playpen where I upload my data and run it, or do I download that kind of the codes, some kind of an executable file and run it on my computer? How does it work? Can somebody explain?
Can I just suggest that again? I'm trying to, I think that's an overarching MMF related question, which you guys, if you could, I think that will be, I think it's a longer conversation and probably it is for the next session. So if you could just table that and come back maybe if there are any kind of specific questions related to the LAI part, because I think we're going back and forth with an overarching MMF versus the modalities. And I wanted to focus on that a bit. Yeah, go ahead, Zhang. Yeah, go ahead.
Just because the models are not completely, I think, as well developed as in the oral space, and we know this from having developed LAIs ourselves, as I work for GSK. And what you just said about generating the model and then establishing a discussion with the agencies on the studies to validate the model, it's almost a discussion, it's a clinical discussion almost. And so are we even ready to talk about MMFs in the LAI space? Because it's very specific again, and there's not as much validation or models out there yet.
Well, if you're talking about a model using PubPK, for example.
Yeah, exactly.
Okay, then it's going to be specific to the drug product.
Exactly.
From the FDA perspective, or from the American public perspective, there's a real benefit of having an MMF, because as we talk about when we do this work for generic companies, and the work takes like, 3 years that the generic company has to spend with us doing the modeling and all of that, proving that the study design will work, etc. It's a lot of work, a lot of money from the generic industry. And so if there could be, all this work could be put in an MMF, and the generic company after that could use that MMF, it would decrease the cost to develop that long-acting injectable a great deal.
I'm actually not
For
debating
the
that at all.
second generic, for the third generic, I mean, it would be immense, the benefit.
Right, I'm not debating that
Okay,
at all.
no, okay.
The question is more around, are we even there yet? I mean, can we be speaking about MMFs for LAIs?
Yeah. No. Yes, we are there. Yeah. I mean, we've presented at previous meetings where, we are there. I don't see any
And the
problem.
reason I
agree.
say that
I
is
really
from our
think-
own personal experience.
It seems like, I mean, of the 27 long-acting injectables that were available in 2021, then there was half of them had models that were describing their PK, so the population PK. So I think we're not. I think we're there. We do have pop PK models that we can use for bioequivalence evaluation.
Okay, and I speak from experience of using, even moving sites with exact same products, having all of the data, the amount of discussion. We were very successful in using the model integrated approach, but it's a lot of discussion yet, and that's why, just from our own experience, it's hard for me to imagine that we are at an MMF stage. That's really where it's coming from.
So due to the time, we will accept one more question.
Yeah, thank you. So there's a technical question on model verification. So actually, starting from Dr. Hooker, you mentioned that if you have like an open source model, someone could change that. Now, if you change the numerical solver or a parameter or equation, you need to verify the model as well, correct? And also, getting back to the previous comment from GSK, I noticed you kind of interchangeably use validation and verification, but they are like separate things, right? So I wanted to have FDA comment on that if the model should be verified and validated, or just verified, or just validated.
Maybe I can comment briefly on that. So there is a ongoing debate that whether it is verification or validation, right? So you will see these wordings or terminology in different guidance in different way. What we say in our own word, validation, you may take it as a verification. So let me make it simple. So what within our division or within our office, what we consider. So when you show the model is capable to predict what it's supposed to do, so it is kind of, we say that it is verification. And validation is when we are assessing your model and it is suitable for the intended purpose. So we are validating the use or your verification. So there is a lot of confusion, I know. Probably you may refer to our guidance. And paper as well. We recently published a paper, I think Eleftherios Shakalogiou on the diclofenac and locally acting PBK modeling. On that paper, we discussed these two terminology more elaborately.
If I might add some points here. So I have a problem with using the word validation in general. You have it in also when IT systems, they say they have a validated IT system, and of course it's just an evaluation. You don't know the truth, right? And that's why you always have bugs in software. You can validate a software, but it's really just an evaluation. You've shown that you get sort of what you expect. And then somebody finds something where they want to test, and they get something that they didn't expect, and that's a bug, and then they have to fix that. So these softwares or models, they'll be evaluated, and at some point you think, that's pretty good, and then you find something that has a bug, and you say, oh, look, we have to change it, we have to update it. So just because something is validated doesn't mean it's perfect and it should never be changed.
I'm just using my past experience when I was at the FDA and OPQ, so writing the guidance for the model submission, and also working with CDRH that they have their own guideline for like verified context of use validation. They also have verification validation 40 with the ASME. So is everything is defined by like CDRH and the OPQ part of the CDR? I was just wondering if OGD has a different approach. And it would be like
I can quickly comment on that as well. That you will see that verification in other guidance that it is used to show that the numerical code and other equation that are correctly incorporated into the model structure. So we are not trying to actually harmonize all the terminology because the context of use, the verification or validation, it can be different from the CDRH and OGD's perspective, like PBK modeling and POP-PK modeling. So these are very different things, right? So I just want to make sure that you understand that there is some differences in the terminology.
Thank you, everyone, and thanks all the panelists to join the panel, and we will take a break now.
It's a very challenging job for me to do, sitting in between you and the break, and also I'm sure the virtual audience may be ready to move on to other things, but I won't take long. But I think it's a very lively discussion. I think a lot of interesting questions bring up. Can you pull, I have a few slides you can pull up. So, so I'm Lei Zhan. I'm the Deputy Director for the Office of Research and Standards in OGD. So it's very nice to have seeing you here in this workshop.
We're just bringing up the slides. Give us a second.
Yeah, I can start talking. So we already know modeling and simulation has been increasingly used in the drug development. You heard this morning from Dr. Pak, the history, the evolving of increasing use of modeling simulation in drug development, not only for the new drug, but also for the generic drugs. You also see the regulatory acceptance for the modeling approach by the formation of Division of Pharmacometrics in OCP, and also 10 years ago, we formed the Division of Quantitative Methods and Modeling in OGD. So to kind of facilitate the development for both new drugs and the generic drugs to using the modeling approach. And we see increasing number of the application to the agency. And also we have tried to assist the industry by providing those meetings to discuss those modeling approach along the drug development pathway. Especially in the OGD, we also have generic drug user fee amendments fee program to do a lot of research, which you heard some of them today, and to see their utility, to assist the industry to use those modeling approach to address the development challenges and try to get the drug faster, approval faster to reach to the patient who may need those medications. So, and you also heard that just a few weeks ago, actually last month, CEDAR established a new center of excellence on quantitative medicine. So the goal is really to, because we do see the modeling used in the drug life cycle, both new and generic drugs, and also the Office of Pharmaceutical Quality. So we want to, in center level, we want to facilitate and also coordinate all these continuous evolution, and also to be consistent application of those quantitative medicine approaches across CEDAR, and also drive the innovation and also integrate those approaches to maximize the patient and the societal benefit. And we also heard, or maybe you're aware, the global harmonization is also ongoing on the model-informed drug development through ICH M15. Actually, we do have some of the members who work on that guidance in the audience. They also comment on the global harmonization. We also heard from one of the speakers from industry also like looking forward to some kind of global harmonization, not just on the maybe modeling part, but also the MMF part, which is the topic for today. And we see this model master file is very an integral part of the model-informed evidence or model-informed drug development. It's happening already, but we may not formally call it MMF, or some concept already be utilized, or the benefit of being recognized, such as we may want to reuse the same model either internally or across, like if it's in the public domain, maybe across the applicants, across the drugs. And also one speaker talked about the object, they treat this as object that could be maybe a precursor to the MMF. But really, we recognize the benefits of the MMF in terms of the reusability to help improve the efficiency, so you can reduce the redundancy to not to have to reinvent a wheel. Then you can leverage the prior knowledge gained to apply to the future application. And we also know the definition, at least from one of the recent publications through the workshop like this, is MMF is a quantitative model or a model platform that has undergone sufficient model verification and validation to recognize as a shareable intellectual property that is acceptable for regulatory purpose. So very important is the regulatory purpose. So you want to drive to the next level. Maybe you can use it for internal decision-making, but you really want to drive to the next level to get the regulatory acceptance and can help your drug to get approval faster. And we also know that MM has the potential to support innovation and streamline the drug development and also improve the efficiency and consistency. And another benefit could be like reduce the risk of the innovation, possible innovation, and also maybe more regulatory certainty to help you to get the drug approved. And I also see one of the important thing of MMF is in the accessible part of the MMF, because they can be utilized by applicants either with or without internal kind of expertise to build those MMF. So this is really can encourage a more use of the model master file. And we also view this could be a framework to have a model sharing and a communication among model developers, industry, and FDA. Model developers could be either the industry themselves, or could be the academia, or could be the CRO, or model platform developer. So it's really a broad community engagement. So we know this discussion, the MMF was the concept introduced about 3 years ago in one of the CRCG workshop in October 2021. That then we have a best practice discussion in 2022, and also there's a paper recently published to summarize the discussion there. And at least those reference there, hopefully you, if you didn't get a chance, also be able to access those materials to get familiarized with what is MMF, what do we envision that could help the drug development. And today, I think really we want to drive from the concept to reality, what reality is. That's the regulatory application reality. And we also know there's a regulatory context of MMF, because MMF is very special regulatory context, follow what the drug master file being used. We can vision this could be a subset of drug master file, or could be something similar to drug master file, but in a very similar regulatory framework. So we know the MMF will be separate from a regulatory application, which means FDA will assess the model separate from the application assessment. And also MMF will not be assessed until there is a regulatory application need that model master file. So it's not like some other modeling exercise you're doing, regardless if you have application or not, you may work on it. And also the regulatory acceptability of the MMF will be assessed separately for every new regulatory application. So the model will be, MMF will be reassessed when there's a new application to use it. So how can we move towards the reality? So today you do hear about, in the first session, define the MMF framework. What is MMF? What types of MMF? Who should develop the MMF, submit the MMF? When should be submitted? When should start a communication? And how, right? There's a lot of discussion around that range. And we also heard a lot of benefits, opportunities through learn from the past modeling experience as well as the case examples. But we also heard about challenges, concerns, questions people may have, how to operationalize this process, and what can we learn from the drug master file experience. And I think maybe you're very eager to go, like, what's next? You know, what I'm getting out of this? What is this informing? I see a lot of skepticism, like, oh, how can we use MMF? Are we ready? So these are very good questions. Hopefully we'll have more in-depth discussion in the next in-person small group discussion. So really, when we talk about moving towards the reality is for us to prepare for the future. You heard from Dr. Leinberger this morning, you know, we are anticipating there will be more modeling submission in the future. We will have some capacity both from the regulatory agency standpoint and from the industry standpoint. You have to pick, choose. You cannot work on all the modeling. I also heard one of the industry person did say they may choose to work on some model, then other model they may leverage the MMF. Maybe that's the direction to go in the future. So we do encourage the implementation of the model master file for regulatory use, and we want us to all understand the benefit, realize maybe it's not for every application, every model. There's so many different models you can use, so many different questions you want to address. So maybe we can, as a community, we can identify which area, maybe the high impact area, maybe more ready to implement, the other area maybe need more work, or maybe just should be in some other realm. It's not mutually exclusive. You can either do the MMF or just do your normal model-informed drug development, or MIE, to submit your application. So, and we also know that, talk about model standardization, understand the model is not static, it's dynamic, we all understand that. And also, but we do want to have a common model validation and the qualification strategy to be able to establish and increase the confidence of utilizing those MMFs. Then we can talk about whether open source, non-open source, confidentiality, legal consideration, regular consideration. I think these are all very good questions should be considered. And we look forward to the further discussion and also collaboration among all the stakeholders to reach a mutual understanding of MMF. Then we also want to advance the development of the best practice for us to implement MMF. Hopefully you all share the vision of the benefit future, but we know there's still the work ahead of us to do. So before I closing, I would like to thank our CRCG team. To put together all the logistics, this nice workshop, and also the working planning committee, especially the DQMM, Lucy and Aliphatheria, they are co-chair of this workshop. And I also like to thank all the speakers and panelists for sharing your experience, your thoughts, your advice, your input. These are all very helpful. And lastly, thank all the participants, both here in person as well as online virtual participants. I really look forward to the next small work discussion in person here, but I also want to let you know there are more discussion topics will be discussed tomorrow. We will talk about the new drug space and also more case example on locally acting products, mainly focused on oral inhaled products and topical to the skin product. So with that, I wish you have a good discussion, good break, and enjoy your discussion. And hopefully we will see what we can come out of this workshop. Thank you.